Hello, everyone, and thank you for joining this month's Bench to Bedside webinar series. I'm Nicole Wilson, Program Manager for the United Mitochondrial Disease Foundation. We're so glad you're here. Before we get started, I would like to remind everybody about MitoShare, our worldwide patient-populated registry. This is the ideal place to help capture the overall journey that our patients experience. So please let your patients know about this important registry today. Save the date for MitoMed 2025 in St. Louis, Missouri, June 18th to the 21st. You can learn more at umdfconference.org. We hope we get to see you there.
Hi, everybody. Welcome to the first Bench to Bedside of 2025. I'm really excited to have you join us today. We have a great lineup of speakers, and I'm Mel. I think it's really exciting to see that we now have so much clinical trial activity taking place inside mitochondrial disease that we have to actually spread this over two Bench to Bedsides.
Agreed. Very exciting and happy 2025, everyone. And that's a great segue into our next talk that is going to be given by Dr. Fernando Scaglia, MD. He's a professor in the Department of Pediatrics and the Department of Molecular and Human Genetics at the Baylor College of Medicine in Texas. Dr. Scaglia is also a pediatric neurologist and geneticist at the Texas Children's Hospital. And he has been conducting this trial for many years now, and I am very excited to hear updates today.
Thank you so much, Anil. Very good morning to all of you. So yes, the title of the talk is L-citrulline treatment of nitric oxide deficiency in MILAS, phase 1 dose finding and safety study. This is a study that has been supported by NAMDEC. It's a pilot project of NAMDEC. We can go actually to the next slide. Yes, so based on previous studies that we conducted prior to this clinical trial that actually focus on the use of stable isotopes in children and adults with MILAS who harbor the M3243A2 gene, MTTL1, we were able to see that both oral arginine and oral citrulline restored impaired nitric oxide production as measured by stable isotope techniques. But citrulline was able to be more efficient than arginine in restoring this impaired nitric oxide production, which we thought that actually could be related to the pathomechanism of metabolic strokes. Can we go to the next slide, please? And therefore, we received funding from NIH through NAMDEC to conduct a phase 1 clinical trial. And these are the inclusion criteria. These were adults 18 through 65 years of age who had to harbor the common pathogenic variant in MTTL1, who had to have an elevated plasma lactate at our baseline visit, who had to score 26 or higher on the Montreal Cognitive Assessment Test, who had to have cessation of arginine, citrulline, or compounds affecting nitric oxide 1 week prior to the study, and have to have a clinical diagnosis of MILAS. Next slide, please. And the primary aim, since this was a phase 1, was safety, and we have to look at the maximum tolerated dose of L-citrulline in 10 consecutive adults with MILAS, starting at 10 grams per day of citrulline and going up to 40 grams. And this would be divided 4 times a day, given for a month, with an additional month of follow-up with 2 safety visits at week 6 and week 8, and with primary safety outcomes that included, looking for dose-limiting toxicities. So all these participants were monitored for the occurrence of adverse events. And the secondary outcome measures was the determination of cerebral blood flow and cerebral blood vessel reactivity using ASL MRI, and then the measure of end-tidal CO2 after the breath holding task. We also measure plasma lactate and alanine at baseline, week 1 and week 4, and measure plasma guanidino compounds by untargeted metabolomic analysis at baseline and week 1 to determine potential arginine toxicity. Next slide, please. And we use a time-to-event continual reassessment method to establish the maximum tolerated dose of L-citrulline, and the study subjects were enrolled one at a time serially, and the dosage for each subsequent participant enrolled was increased or decreased based on the determination of the time-to-event continual reassessment method algorithm, which took into account the rate of safety outcomes encountered by previously enrolled subjects to determine dosage for subsequent participants. Next slide, please. So this was actually the table of assessments. During the first month, participants actually had to be be reassessed weekly in our clinical research unit, and then during the second month, when the administration of citrulline had ceased, then they were coming for 2 safety visits at week 6 and week 8. Next slide, please. So these are the baseline characteristics of the 10 subjects enrolled in the study. We actually had no screen failures. There was a mean age of 37 years, 5 males, 5 females. There were 1 participant was African American, 9 participants were Caucasian. The mean for the Montreal Cognitive Assessment was 28. All participants had to have a normal liver function test and normal renal function, in addition to a normal complete blood count at the baseline visit. Next slide, please. These were the dose levels, the time of follow-up, and the presence or absence of those limiting toxicities for each subject. So we started at dose level 2 on the first participant at 10 grams daily citrulline. A third participant was on 20 grams of citrulline. Participant number 3 and participant number 4 on 30 grams, and then the remainder of 6 participants reached the maximum dose of 40 grams of daily citrulline. Participants number 2, number 4, number 6, number 9, and number 10 had adverse events, but there were no severe adverse events and no dose-limiting toxicities. Next slide, please. These are the adverse events that were observed that included headache, nausea, and vomiting, which we deemed to be possibly related, but there was no action taken with the study treatment. The treatment was not interrupted and continued because this self-resolved. Next slide, please. We are not presenting descriptive statistics because of the limited number of participants who actually were enrolled in the study, but we did see a trend, a decrease in plasma lactate levels when we compare baseline, week 1 and week 4. Next slide, please. There was also a trend in decrease in plasma alanine levels when we compare, and here we are only, as the previous slide, we are only looking at the maximum dose of citrulline of 40 grams. There was also a trend decrease in the plasma alanine level. And in the next slide, we do see that by looking at untargeted metabolomic analysis, we were evaluating plasma guanidino compounds at baseline and week 1. This, since we expected that with higher doses of citrulline, there would also be generation in renal tissues of arginine, and guanidino compounds are derived from arginine. It was a way of looking at potential arginine toxicity. This is a semi-quantitative test, and we usually reported with z-scores. There were no analyte outliers with z-scores above 2 that were found. On the next slide, we actually look, so here what we are looking at is the numbers of cerebral blood flow in terms of like milliliters per 100 grams of brain tissue per minute by using ASL MRI. And we look at the 6 participants or 6 subjects who were actually on the highest dose of citrulline, 40 grams. And then we look at different sections of the brain. I think that for the sake of time, I'm just presenting whole brain and occipital areas. In terms of like the baseline values, usually the baseline values that actually are observed by our colleagues in neuroradiology, in let's say subjects who actually do not have this condition is around like 50 milliliters per 100 grams of brain tissue per minute. So there seemed to be a decrease in the baseline cerebral blood flow, and we did see an increase, but more so in the occipital lobe when we compare even to the frontal lobe. We have that data, but it's not in the slide. So CBF increase in the occipital region in 6 research subjects on the highest dose. And then we recently were able to obtain cerebrovascular reactivity. And in the next slide, I'm presenting, we have like images. So we can say this, that actually the cerebrovascular reactivity was able to be determined in 9 out of 10 participants. For technical issues on participant number 9, we were not able to obtain that, but there was an increased cerebrovascular reactivity following a parieto-occipital pattern. And actually, there are other participants that also have an increased cerebrovascular reactivity following other patterns, including like a parieto-temporal, and that's also seemed to correlate with the areas where the metabolic strokes or the stroke-like episodes had occurred. And again, this is by looking at the highest dose of 40 grams of citrulline. So and now in the last slide, we'll see that this was a safety study, phase 1. There were no SAEs, no DLTs observed with any of the used L-citrulline doses, and targeted metabolomic analysis did not show the presence of guanidino compounds with Z-scores of equal or higher than 2. Research participants on the highest dose of citrulline were observed to have improved cerebral blood flow in the occipital region, while on citrulline supplementation, in 9 out of 10 participants, we were able to determine cerebrovascular reactivity and seemed to be increased, mostly in a parieto-occipital pattern during or while on citrulline supplementation. In the last slide is an acknowledgement slide, and as Dr. Pine mentioned, it really took a village to accomplish this study, and I want to thank you for your attention. Thank you so much.
Thank you so much, Fernando. So for the patients listening in today, very briefly, should if they go and ask their doctors to be on 40 grams of citrulline, should they be doing that, or should they be waiting for the next phase of this trial?
No, I think that actually we should be waiting for the next phase of the trial. I think that is too premature to be recommended that. And in reality, although we were trying to determine a maximum tolerated dose of citrulline, we can't really say that 40 grams is an MTD because, you know, we didn't really have a dose-limiting toxicity or a severe adverse event. And these issues, these AEs, self-resolved. Yeah.
Thank you.
But we should wait. Yeah.
Good. Our next speaker is Dr. Jan Smeitnik, MD, PhD. He's the Chief Medical Officer and the co-founder of Khondrion, a biopharmaceutical company dedicated to developing therapies for mitochondrial disease. Jan, please take it away.
Yeah, thanks very much, Amel, for the kind introduction. Good afternoon and good evening, actually, from the Netherlands. Happy New Year to you all. As we step into a new chapter, today we celebrate our achievements and look forward with hope and determination to what lies ahead. May I have the next slide, please? So Khondrion is a phase 3 readiness Netherlands-based biotech company, and backed by decades of scientific and clinical expertise, our in-house drug discovery programs, focus on innovative solutions for primary mitochondrial diseases. At the forefront of our effort is sonlicromanol, an oral, brain-penetrant drug with a scientifically validated triple mode of action. Our initial target population are primary mitochondrial disease patients with the 3243A2G variants, including those clinically diagnosed with classic MELAS, maternally inherited diabetes mellitus and deafness, and mixed phenotypes. And this therapy so far has shown great promise in completed clinical trials and in the ongoing named patient program with now over 2.5 years of tolerability and safety data. Have the next slide, please. So the milestones achieved in 2024 further underscore our commitment to advancing patient care. Results from the phase 2b program published in Brain validate sonlicromanol's positive effect on the quality of life and functions of 3243 primary mitochondrial disease patients. A 70-week follow-up study affirms its long-term safety, while the breakthrough discovery of a new biomarker correlates disease severity with therapeutic effects. Strategic collaborations, such as with the UNDF Mito Fund, expand the impact of our work, and the extension of the named patient program in the Netherlands in 2025 ensures continued access to this promising treatment. With FDA IND clearance secured, sonlicromanol is poised to enter a pivotal phase 3 registrational trial. Have the next slide, please. The impact of sonlicromanol of patients is profound. As patients recently reported, it alleviates their most burdensome symptoms, restores energy, strengthens resilience, and significantly enhances quality of life, as shown in this slide. May I have the next slide? Following 78 weeks of sonlicromanol, patients feel better, are more energized, and are stronger, as evaluated by a variety of clinical outcome assessments. The results show chronic and durable improvements over 78 weeks, which embodies the potential of this treatment to really transform lives. Have the next slide. Looking ahead to 2025, sonlicromanol is ready for its next leap, a randomized, placebo-controlled, phase 3 clinical trial for adults with the 3243A2G mutation. Administered orally at 100 milligram tablets BID, this trial represents a critical step toward making this therapy widely accessible to those in need. Have the final slide, please. Finally, I would like to thank all patients and families who contributed to these studies, mitochondrial specialists, other stakeholders, and importantly also the UNDF for their unwavering support and belief in our mission. Together, we are advancing forward a future where scientific breakthroughs become lifelines and hope is restored for patients with primary mitochondrial disease and their families. Let's continue to move forward, inspire each other, and transform life. Thank you very much.
Thank you so much, and congratulations on your program, and good luck in 2025. The next two presentations are going to touch on ongoing clinical trials, so that's very exciting. Our first presenter is Chad Glatzer. He's a doctor in pharmacology and the Senior Director of Clinical Research at Tisento Therapeutics, a biotechnology research company specializing in rare diseases, mitochondrial diseases, and MELAS specifically. Chad?
Thank you, Amel. As was mentioned earlier, it's exciting to be a part of a totally separate session devoted to all the work that's been done in MELAS. We're excited to be here and give an update on our clinical trial called PRISM. Next slide, please. So before we talk about the study, for those of you that aren't already familiar with Tisento and our lead program, I have a couple slides to provide a bit of background. We were founded to continue the development of zigosogwot, prioritize it as our lead program. and evaluate it as a treatment for patients with MELAS. It works on the nitric oxide pathway, so it's relevant to the presentations that were given earlier in the session. It's an SGC stimulator, so it works similar to the other SGC stimulators that are approved. However, it is able to get into the brain, so it's brain penetrant and can modulate this pathway centrally. It's oral, it's given once daily, and it is basically amplifying and restoring deficient nitric oxide signaling, of course, not only in the periphery, but most importantly in the CNS, which is a key aspect of the mechanism, given that we're targeting MELAS, a very neurologically focused phenotype. In terms of how this works and our belief that this is really the most optimal way to modulate this pathway, it's leveraging the body's natural endogenous on-demand production of NO and the signaling associated with that. It's also binding to the central node of the pathway, SGC, and increasing cyclic GMP concentrations via that mechanism. You can see the therapeutic effects here. We already heard a little bit about some of the effects of arginine and vasodilation and the cerebrovascular hemodynamics associated with this pathway. But again, there's direct impacts on mitochondrial function, things like mitochondrial biogenesis, as well as impacts directly on neuronal function. Our hope is that this kind of multi-pronged approach and broad pharmacology will lead to decreased fatigue and improved cognitive function, which we'll talk a little bit more about on a subsequent slide. We do have data from a prior phase 2A study where we confirmed target engagement and aspects across each of these physiological domains. Next slide, please. So I think I've shown this slide in every presentation that I've given since we were founded, but I think it's important because it captures kind of our thought process moving from a disease that is challenging to evaluate in a clinical trial to a protocol that we feel very confident in, that we think is really fit for purpose and able to measure zigosogol's efficacy. We started with understanding the disease burden. We conducted a patient interview study to identify the patient perspective of the symptoms that are most important to them. We also incorporated expert perspectives into that trial or that study. We used all that data and mapped it and used it to select, outcome measures, both PROs as well as performance outcome measures that we think are able to capture those concepts appropriately. And then, of course, incorporated those into our study with the aim of establishing a treatment effect and determining what our dose and what our approvable endpoints would be for a subsequent study. Next slide. We don't have time to go through the results of the qualitative interview study, but I did want to show this slide, which I think is one of the key takeaways from that trial. So, we enrolled patients that had MELAS. Other studies that have looked at the symptoms of mitochondrial disease did not really focus on MELAS or patients with stroke-like episodes. So these are kind of the frequency of concepts that came up most frequently in patients as well as clinicians. And they really fall into two buckets. I think fatigue isn't a big surprise, that there's a number of aspects of fatigue that patients are reporting: physical fatigue, mental fatigue, exercise intolerance, weakness, muscle fatigue. But there's also a central component to this, and cognitive deficits, again, given that they've had a history of stroke-like episodes. Mental fatigue is kind of a cognitive symptom as well. There's issues with executive function and processing speed that we know. There's memory problems, difficulty concentrating, difficulty finding words, and then, of course, a number of other CNS symptoms. So these are all the things that we took into account when we prioritized our endpoint model for our study. So this is our trial. It has been initiated. We do have active sites. It's a randomized double-blind placebo-controlled study. The most defining feature is that it is a crossover trial. So there's two distinct 12-week treatment periods separated by a 4-week washout in between. We're evaluating the same dose that we evaluated in the prior phase 2A study, the 50 milligram dose, but we saw some effects, concentration-dependent effects that made us want to evaluate a higher dose. So we're looking at 30 milligrams as well. The eligibility criteria of importance are adults. You have to be at least 18 years of age. We're focused on mtDNA mutations, so not just 3243, any mtDNA mutation, provided that the patient does have a history of stroke-like episodes. So there needs to be at least one MRI showing a stroke-like lesion, so we can feel confident that the clinical phenotype is due to that pathophysiology. The primary endpoint model is focused on fatigue and cognition, going back to what we heard from patients and experts and what we believe really makes up the disease experience from the patient perspective. Fatigue, we're using a MELAS short form that has been adapted from actually what Magnus and Abliva has done to incorporate a little bit more of a mental fatigue component to that, given this study population. So that's a PRO. And then we have two cognitive performance tests that we're really excited to incorporate. One that's focused on executive function and one that's focused more so on processing speed and attention. Kind of getting at the mental fatigue aspect and some of those other challenges that patients are reporting. All of these will be combined into a single statistical test, which does a number of things. It hopefully justifies our claims that we're impacting multiple aspects of a complex disease, but it's also a way to overcome some of the heterogeneity of the population where there's not one outcome measure that is, it's not a one size fits all. And it also increases our power with fewer subjects. Safety and tolerability, of course, is a key aspect of the study as well. So we'll be looking at that. And 44 participants is our target approximately. It is a global study, US, Canada, UK, Italy, Germany, and Australia. And I'm happy to say we have been screening patients. We have a lot of baseline data that we've been looking at. And I think it really increases our confidence that these outcome measures are able to assess cognitive deficits and the overall disease deficits in this population, and hopefully makes them fit to assess efficacy of zigosogod in this population as well. That's all I have. My contact information is here. If you're interested in the study or our program, please visit our website, tisentotx.com.
Thank you very much, Chad. And last but not least, Dr. Magnus Hansen. He's an MD, PhD, Chief Medical Officer, and Vice President of Preclinical and Clinical Development for Abliva, a biopharmaceutical company focused on developing treatments for primary mitochondrial diseases. Magnus?
Great. Thank you so much, Shamel. And thanks to the UMDF team for arranging this session. It's really exciting to hear all the updates from the different companies and researchers trying to develop new treatments for these devastating diseases. So we can move ahead with a slide. These are just the usual disclaimers. You can go to the next one. So I actually wanted to start with highlighting some news and recent developments. So Abliva is focused on developing new treatments, pharmaceutical treatments for mitochondrial disease. Our lead program is KL1333, and this is now in a late-stage clinical trial called the Falcon study. So this study is what we hope to be the last placebo-controlled trial to seek regulatory approval. And we designed this trial with an interim analysis. So last year, we had the most important milestone achieved for this program and the company so far. We passed successfully an interim analysis. So in the interim analysis, kind of like a halfway test into the trial to check if everything is on track. And it was. So specifically, we tested safety, or an independent data monitoring committee evaluated the safety, and we also did futility assessments of the two primary endpoints, and they both passed. So meaning that there is some signal of efficacy in the trial. So that is, of course, super exciting for us. And it generated quite a lot of interest, and that leads me to the second major update. So right before the holiday, the shareholders of Abliva had an offer to be acquired by another company. This company is called Pharming, and it's a larger company than Abliva. So that could actually bring more resources to the program, which we hope would facilitate an efficient completion of this program. So the study is planned to continue in 2025. And if we go to the next slide, there's another piece of news as well. In this January issue of the journal Brain, we published the first patient data with KL1333. So this is one of our 3 phase 1 studies, the study that included patient data. So this is the paper, and I think, yes, there is a link in the chat if you're interested to read more. It's a nice introduction to KL1333, and it's open access, so everybody can access it. So if we move to the next slide, a little bit more about the Falcon study. So what is this study about? Well, it is a trial to evaluate the safety and efficacy of KL1333. And KL1333 is a NAD plus NADH modulator, which we think is potentially very important for the energy regulation in mitochondrial disease. So the aim is to normalize that ratio and thereby improve the energy regulation, both short-term and long-term in mitochondrial disease. If we move to the next slide, some specifics about the Falcon study. So this study is actually a little bit broader. It doesn't only include MELAS or M3243A to G patients, but also other mitochondrial disease. It has to be caused by mtDNA mutation, definitely pathogenic mutation, and be associated with multisystemic disease, specifically chronic fatigue and myopathy in the lower extremities to match the study endpoints. If we move to the next slide, it's about a year-long study, a bit more than a year, and there is placebo-controlled treatment for 48 weeks. So meaning that the participants would either receive tablets with KL1333 or visually identical tablets but lacking KL1333. Treatment is for 48 weeks. Before that, we have a 2 to 3 month screening period to make sure that we have the targeted patient population, and then there is a safety follow-up. And the next slide, you can see how the tablets look like. It's small, round, flat tablets. They should be quite easy to swallow. And in the study, participants take either 1 or 2 tablets in the morning and 1 or 2 tablets in the afternoon. And it's evaluated according to the maximally well-tolerated dose. And 60% will receive the active compound and 40% the placebo. And of course, it's blinded, so nobody will know which treatment is which until the study is fully completed. If we go to the next slide, so the study, as I mentioned, is focusing on fatigue and myopathy in the lower extremities. And we have 2 alternative primary endpoints. And that means that if either or both of them are show benefit, that's enough for the study to be considered successful. And Chad mentioned the PROMISE fatigue short form. So we developed and validated this short form based on the PROMISE item bank a few years ago. And the idea was that should be widely used in the mitochondrial disease research community. So really happy to see that has actually happened. And the second endpoint is a test of the muscle function that we follow throughout the trial with the 30-second sit-to-stand test, where we ask participants to rise up from a chair as many times as they can during 30 seconds. If we move to the next slide, so to the right here is the ongoing Falcon study. We did a number of phase 1 studies, including the phase 1a/b trial with patient data, which is in the Brain publication, but also the validation of endpoints and collection of natural history data, which is continuing. But that led us to the design of the Falcon study, which hopefully is the last placebo-controlled trial we need for KL1333. We have completed the first wave of recruitment. So after we had recruited 40 patients, we followed those patients for the interim analysis, which, as mentioned, was successful. And that data has now triggered and enabled us to prepare the wave 2 of the study. And that will include an additional 140 patients that will be recruited globally at about 40 sites, including roughly 10 sites in the US. And we expect that recruitment to be reinitiated now in early 2025. So with that, I would just like to thank you for listening and hand the word back to you, Emil.
Thank you so much, Magnus, and thank you so much to all our speakers today. Those were wonderful updates, and we're very excited to be hearing more and more of these updates to see how excited the community is about mitochondrial diseases and finding a therapy for these patients who desperately need them. Phil, I'll turn it back to you.
Thank you, Mel. Thank you for your able moderation. Appreciate that and underscore the deep appreciation to all our speakers today, Dr. Tyne, Dr. Scaglia, and our three industry representatives. It is an exciting time for the community and really look forward to continued updates, including at MitoMed 2025 in St. Louis, where once again, we'll bring together the entire community and make sure that clinical trial updates are a component of that program. I'm just going to wrap up with a reminder that Bench to Bedside is a monthly series, and in February, we have a focus on the role of rehabilitation medicine in the care of children with mitochondrial disease. Several speakers you see listed on the screen will be joining us. So that's Monday, February 10th, 2025 at 12 p.m. Eastern Time. With that, we're going to wrap up today's Bench to Bedside. Thank you for your attendance and look forward to seeing